Go ahead and do a brief introduction.
So I think I talked about the Copal lab.
Yes. Okay. Back and-
Was there a question?
Oh, no, you can keep going.
Oh. And the next thing I'm going to show. How come it's not advancing?
Yeah.
There you go. So this is in a nutshell how the specimens we're going to go through. I'm going to talk about vertebrate eyes. If there's time at the end, and if people are still interested, we also have a fairly good collection of invertebrate eyes, and I can transition to talking about invertebrate eyes. But for most of this, the goal of this course is to talk about the comparative anatomy of the vertebrate eye. You'll see at the bottom, it references a website called Time Tree of Life. Is there anybody here that's familiar with that website? If you're interested in phylogeny, you can just type in a Google search for Time Tree of Life, and on that site you can compose a phylogeny. I put this together myself. You can put down two different taxi, like you can say human or Homo sapien and robin or cauliflower, and it will calculate using scientific data that's available how long ago there was a common ancestor between the two taxa, or you can put together a group like this. With that side-by-side comparison, you can use common names. It's tricky to use common names. What I like to do is go to Wikipedia, where we can use common names, and then Wikipedia will tell you the scientific terminology for the taxa that you're interested in, and those scientific terms usually work well in the Time Tree of Life. So you can put those in. Or you can put together a phylogeny like this. It's the way to do it is, if you're interested, is to, rather than putting in something like monotreme, put in the scientific term for duck-billed platypus, for turtle, get the name of a turtle, and then put that in the scientific terminology. And then it'll do a search, and it'll print out a phylogeny like this, along with the paleontologic epoch from which the animals diverged. So this is a phylogeny for the vertebrates. It starts back even before the Cambrian. On the left of the C there is for the Cambrian period. And the most primitive, the most basal of the things that we're interested in would be the jawless fish, and the extant jawless fish are lampreys and hagfish. And then next would be the sharks, and next would be the rayfin and lungfish. And you can see, if you trace the lines back, you can see how many million years ago in the very bottom, or what paleontologic epoch there was a common ancestor. We'll probably be spending most of our time with the reptiles and mammals, and you should already all be aware that the birds, phylogenetically, the birds come from the two-legged carnivorous dinosaurs, the name I can't remember, the theropod dinosaurs. So birds are to the phylogenetic classification are reptiles, and you can see where they stand. Birds, of course, all the dinosaurs are gone, but also already split off from the rest of the reptiles in the dinosaur group are the crocodilians and the turtles. And then more primitive than that, we have your lizards, and we'll talk a fair amount about lizards. The tuatara is an extant animal, which is it's a reptile. It's very ancient in terms of its derivation. It doesn't match with the lizards. It's more ancient than that. And I don't think I have a copy. We have a slide of a tuatara. I may mention it. I don't think I have one in the slides that I'm going to be showing. Any questions about phylogeny? And I'm going to try and to the extent that I can, I'm going to try to superimpose the anatomy with the phylogenetic origin, and we'll come back to it periodically. The other the other pattern that we'll be talking a lot about is the comparative anatomy of the eye relative to the niche that the animal exists in. Next. So I've chosen to start with the most complex eye, with the eyes that has the most anatomic complexity and the highest degree of visual acuity, and those would be the birds. And then we'll go from there. We'll talk about some reptilian features, and then we'll phase, and then we'll sort of go back and forth phylogeny and niche to kind of fill in the blanks. This is a red-tailed hawkeye. For the purposes of this discussion, there's nothing special about it being a red-tailed hawkeye. There are things that are special about the red-tailed hawkeye, but we're not going to talk about them right now. But we're going to talk about some of the features of the bird eye. First thing to notice is that the shape of the eye has, I call it a pear shape. Can anybody tell me why the bird eye would have a pear shape? So before we get into that, in order for the eye to function as an optical instrument, it has to be rigid. You can't have an optical instrument that's floppy and changing shape all the time. Can anybody tell me how it is that the eye acquires its rigidity? There is a scleral ossicle in a portion of the sclera, and that in fact would be rigid, but it's only a small portion of the. So you speak loud and clearly. And what is it about the liquid in the eye? It's you're right on.
I'm super new to the eye, sorry.
It's the liquid under pressure.
Oh, yes.
I think that's what you meant, but
Yes, we actually research on that.
yeah, it's the eye maintains its rigidity because of the pressure. The liquid in the eye is maintained under pressure. And so if you have rigidity based on pressure from the liquid in the eye, then you're going to have a spherical eye, or you might have a little bit of change in the curvature of the cornea, but it's going to be basically spherical, like almost all mammal eyes are. The bird, on the other hand, somebody mentioned it already, has a scleral ossicle, a bone in the eye, basically right about here, and you can see that bone goes circumferentially around the eye, and it compresses the front of the eye, so it allows the bird to have this pear-shaped pear shape. And so the advantage of the pear shape, in terms of visual acuity, the important dimension of the eye is this axial dimension from the front of the cornea, from the axial cornea back to the retina. That's where the focus occurs. That's where the focused image on the retina occurs. The other dimensions are not as important. But one of the advantages of having this pear-shaped eye is it will maximize this axial diameter without adding extra weight that would be required for the spherical eye. And if you think of a bird, on the first image there was a picture of a dissected owl head, just to show you how big the eyes are relative to the head of a bird. For a flying bird, it's a lot to expect it to have a big sophisticated optical structure in its head. Okay, probably the next thing to talk about is this thing that jumps out at you here, this structure that looks for all the world like a radiator that's sticking out of the back of the eye. Does anybody know what that is? That's called
The pectin.
pectin oculi. It is a vascular structure. We'll go over the histology of it a little bit.
Do all birds have pectin?
I believe all birds have a pectin.
Okay.
I'm not completely sure that the what's the flightless bird from New Zealand?
Kiwi.
The kiwi. I'm not completely sure that the kiwi does. I have a kiwi that I'm gonna show you. I don't think the section shows the pectin, but it's stick a pin in it because the kiwi may not have a pectin. In most birds, it has this radiator shape, and it's always when you look at the gross image of the eye, it's always very rigid, and even when you section it to get a histosection, it's usually very rigid. And the only reason it has rigidity is because it has this architectural structure of being plicated and then held together on the top. The retina of the bird has no blood vessels. In fact, if you're going to find an animal that has blood vessels in the retina, then it's going to be a mammal, because no other vertebrates have blood vessels actually in the retina like a human does. And then there's a whole bunch of mammals that don't have blood vessels in the retina as well. Horses, guinea pigs, a whole group of rodents related to guinea pigs, elephants, and then animals related to horses like rhinoceros and tapirs, and then a bunch of more exotic mammals. So a whole bunch of mammals that don't have blood vessels in the retina. The take home from that to me is that the inner, and they would provide nutrients to the inner retina from the pectin by diffusion across the vitreous. And the take home for me is that the inner retina, the nutritional needs of the inner retina is not nearly as important as the nutritional needs of the outer retina. Well, I'm not going to talk much about it here because you really can't see the structures, but the photoreceptor cells are the most oxygen-dependent cells in the body. And we'll talk about the blood supply that supplies the outer retina, the photoreceptors, when we start showing the slides. Notice the lens is kind of funny shaped. It breaks off into a different appearance at the equator, and we're going to talk about that. That structure is called the annular pad, and it's probably better talked about when we talk about the histology. And so I think that's all I wanted to show this because the structure of the pectin is more clear when you look at it as a gross structure, and the overall shape of the eye is nice to look at with this specimen. Okay, so let's go to this microscope slide. So this is a pelican. And there's nothing special about it being a pelican, except that it's a nice, well-sectioned, anatomically correct representation of a bird eye. And we can see a lot of the same things we already talked about. Here's the pectin oculi. Now you can see once again the annular pad of the lens. There's another feature that people should be aware of in the bird eye. Does anybody know anything about the musculature of the bird eye? The bird, and not only the bird, but all reptiles, and only reptiles, have skeletal muscle in their eye rather than smooth muscle. And that's going to be important in a number of ways, which we'll talk about very shortly. Yeah,
So that little structure, what is it, the pectin oculi?
yeah.
It looks like it's coming right off the optic nerve.
It comes right off the optic nerve, right off the inner side of the optic nerve. It derives its blood vessels from the choroid, but it's positioned right off the optic nerve, which of course would be a blind spot. Would be a good place to put something that would interfere with vision.
So is it coming off the same artery that would be?
That's a great question, though. Arterial supply of the pectin, don't know. So here you can see this. This is the scleral ossicle, the bone, and it's in a bird. It's actually several ossicles that are kind of tied together, and they wrap around like a belt at the level of the limbus, where the cornea meets the sclera. And then you can see this annular pad here at the equator of the bird lens. So let's use a little more magnification. Here's our pectin. And if we go and zoom in on it a little bit, I'm going to zoom in a little bit more. You can see, as I've said already, it's basically a tangle of blood vessels and little else. It's got some pigmented cells, but it's a tangle of blood vessels. Like I said before, there's nothing about this tissue that would provide it with rigidity, except for the architectural structure of the way it's held together. Now I'm going to switch from this one. I'm going to show you a.
Do you have any idea where the pigmented cells come from?
They're neural. All the pigmented cells are neural crest, as are the pigmented cells in the choroid. I do not have any idea whether the pigmented cells in the pectin come from a different derivation than the choroid or the anterior uvea. The pigmented cells in the epithelial cells are neural tube origin.
Right.
So I've already shown you a gross picture of the pectin. You realize that little squiggly worm doesn't do the structure of the pectin justice. But this now is a hawk. It may be the same hawk that we just looked at. This is a histosection of a hawk eye, showing the pectin again in all its glory. And you can see it's a plicated like a Japanese fan, and then it's held together at its apex by a solid bar, and hence the rigidity. Okay, now let's talk about the vascular supply of the retina. Oh, another thing before we go any further is you should notice that the bird eye, the sclera of the bird eye has a layer of cartilage. So not only does it have bone in the eye, it also has cartilage. And once again, if you have an eye, a vertebrate eye, and it doesn't have cartilage, then it's either going to be a mammal or a snake. All of the other vertebrates have cartilage in their eye. And I use the word, you should always be skeptical when someone uses the word all, because there are exceptions to that rule. If you get into really primitive cave fish and things like that, there may not be cartilage, but mammals and snakes. And keep that combination in mind, because we're going to talk about it several times. And hopefully I'm going to explain why. Another thing to notice. So this is the choroid. And if you're sharp-eyed and you understand what you're looking at, you'll see that this choroid is just filled with red blood cells, way more than one would expect of physiologically. And I'm not completely sure I know why that is, but I have a strong suspicion. And my suspicion is that this animal, and we see this in horses sometimes too, that this animal was euthanized in the postmortem room and then held by its feet upside down so that the head dropped to the bottom and all the unclotted blood rushed to the head. And I think that's why we have all this blood in the choroid. So don't worry about that. Again, no blood vessels actually in the retina. So the blood supply, proteins, amino acids, oxygen, reaching the inner retina is going to have to diffuse through the choroid from the pecten. There's a lot of general ocular anatomy that I'm not going to dwell on. So if you're expecting me to go through the 10 or 11 layers of the retina now, I'm not generally going to do that. If you're expecting me to talk about the physiology of aqueous production in the ciliary body, I'm not going to do that. I'm going to stick to features that stand out as being important in comparative anatomy for particular niches of different animals. So the some reason I can't get that in focus.
Use the fine. There you go.
So the inner inner retina includes the nerve fiber layer, the ganglion cells. Perhaps it goes as far as these layer of nuclei here called the inner nuclear layer. The photoreceptor cells are the nuclei of the photoreceptor cells are here. That's the outer nuclear layer, and then the rubber hits the road where the photoreceptors work on the outside of the nuclei, and where they have the inner and outer segments of the photoreceptors, and that's where the oxygen consumption of the photoreceptors is most important. So this mechanism of diffusion of materials from the pecten oculi to the retina isn't going to work. It's not going to be anywhere near as good enough to supply the retina and the outer retina. So the blood supply to the outer retina, of course, comes from the choroid, and it comes from a particular capillary structure called the choriocapillaris. And just as the photoreceptor is the most oxygen-dependent cell in the body, the choriocapillaris is the most heavily perfused capillary bed in the body. Unfortunately, once the heart stops beating, the blood in the choriocapillaris leaches out, and the pathologist is left scratching. We no longer are able to really see the choriocapillaris except if I went to high magnification, I could show you some things and you'd all go away completely underwhelmed in terms of the function of the structure of the choriocapillaris. Fortunately, I'm going to show you some examples where it stands out a little bit. But before I do that, I'm going to point out one other feature of the retina. And that is the relationship between the retinal pigment epithelium. That's those pigmented cells here. And this pigment, the retinal pigment epithelium is from the neural tube, as opposed to the neural crest. And so these are neural cells, and they're pigmented. If you're used to looking at the pigmented epithelium of a dog or a cat or a human, I think hopefully you'll be impressed with this specimen, that the pigment is A, more robust, and B, it actually interdigitates with the outer segments of the photoreceptors. Let's go to high magnification and just see if we can demonstrate that. The inner segment of the photoreceptors is from here to here, and that contains a rough endoplasmic reticulum for making proteins. It contains a whole bunch of mitochondria because, as I said, this is the most oxygen-dependent cell in the body. Then it transitions into this structure here, which is a modified cilia, and that's where that's where the light is transformed into electrical energy to initiate a signal that ends up going to the brain. And that outer segment is embedded in the pigment from the retinal pigment epithelium. I'll show you some perhaps better specimens, but I think even with this, if you're used to looking at dog, cat, or human or mouse retinas, I think you can see how robust that pigment is and how it actually migrates up and shades the photoreceptor outer segment. So I call this phenomenon the shading of the photoreceptors. And once again, you should expect to find this phenomenon of shading of the photoreceptors in every vertebrate taxa except mammals and snakes. And stick a pin in it because there are some exceptions to that rule as well. Can you think of what the advantage to the animal would be if you shade the outer segments of the photoreceptor where the light actually interacts with the tissue? Why would you want pigment in the eye altogether? What's the benefit of having pigment in the eye?
Could be that birds are always really high up in altitude, so they get more UV exposure potentially, so you need more pigment for protection.
Birds are able to tolerate bright sunlight, possibly because they go high up in the air, but also just sitting on branches and what have you. So you're sort of on the right track. What would be the advantage? Let's say it's to prevent vision loss due to bright sunlight.
It prevents reflections.
Yeah, it prevents glare. It prevents the light in the eye from bouncing around the eye and stimulating photoreceptors that it's not meant to stimulate. Now, in a mammal, how is the same thing accomplished? You're in bright light. Right, there is pigment in the pigment epithelium, but only to the most minimal extent does it actually shade the photoreceptor. Yeah, the iris constricts. So in bright light, your iris will constrict, limiting the amount of light that can get to the eye. I'm going to talk about it here, and we'll come back to it later. I've already said that birds have skeletal muscle in their eye, including both the iris and ciliary body. And the fact that they have skeletal muscle in their eye means that their iris sphincter is under voluntary control. And they can do that because this mechanism is very effective in preventing glare in the eye. Just the other day, we have a parrot, and whereas my cat, she was sitting with the sun coming through the window directly onto his face. And the cat, same thing, had the most minuscule slit pupil you could imagine. I looked at my wife and she had a very small pupil, but my bird's pupils were still considerably large. And they can do that because of this protective effect of the shading of the photoreceptors. That means that they can use their pupil, flashing their pupil back and forth for nonverbal communication. And I've learned a long time ago that if my bird was flashing its pupils at me when I put my finger out, that I should not offer him to sit on it because it means that he's agitated and he's going to bite. I have many scars to testify to that. So you can kind of get a hint here of the choriocapillaris. I'll show you a better example of that. But it's a capillary network. Once again, the most heavily perfused capillary network in the body. Bird eyes are adapted for visual acuity. If you're looking at an eye histologically, the way to tell if you're looking at a part of the retina that's adapted for visual acuity, look for the number of ganglion cells. This is a moderate number of ganglion cells in a bird. It's nowhere near the center of visual acuity. And now I'm going to switch to a camel eye. Seems like a big switch, but I know that this camel eye has a choriocapillaris that's visible. So this is a huge eye. We'll just attempt to show you how huge it is. We'll go back down to here. I'm not gonna try to adjust this, but you can see how compared to the pelican eye. So I put this on the scope. And we're gonna zero in on the retina. And I have no idea why the choriocapillaris. I think it's better in a different area. In this camel, it's still not so good, but you can see the open lumina of the capillary spaces. I'm gonna see here's a better area. I have no idea why in this camel eye compared to most other eyes that we have the choriocapillaris is so visible. You can see that it's heavily pigmented. The camel has a pigmented retinal pigment epithelium everywhere, but the pigment does not shade the outer segments of the photoreceptors. Instead, it does provide protection against glare for the same reason the insides of a camera are black, but it doesn't shade the individual photoreceptors so that light would reach one and only one photoreceptor. We might come back to this camel eye. It has some other features important for vision in bright light. Okay, back to the pelican. I'm going to transition to some other features of the bird eye. And then we're going to come back to the retina, particularly to talk about visual acuity and color vision. So here we are. We're in the back of the eye where we were. We're going to come around here to the front of the eye. You can see that the scleral cartilage stops. And about the area where the scleral cartilage stops, we have the scleral ossicle. We have this bony structure in the eye, which forms a belt around the limbus, causing that pear shape, causing the eye to have that pear shape that we talked about already. So now we're going to look at the ciliary muscle. And I've already mentioned that it's skeletal muscle. And so here's a portion of it. And here's another portion over there. I like to think of the ciliary muscle of a bird as being anterior ciliary muscle and posterior ciliary muscle. If you get a book or you search on the internet, you may well find that the bird has 3 ciliary muscles. Believe me, I've spent a lot of time looking at a lot of bird eyes trying to identify 3 ciliary muscles. And I can usually identify these 2, but I almost never can identify anything that I would call a third. So I just abandoned that in terms of my terminology. The anterior ciliary muscle is called Crampton's muscle, and the posterior ciliary muscle is called Brooks muscle. Now we're going to start with these muscles, and we're going to talk about the phenomenon of accommodation. Does anybody know what, if I'm talking about accommodation, what I'm talking about?
Focusing.
Yeah, focusing on things that are either near or far. Basically, when you're using your muscles to accommodate, you're trying to squint your eyes into being able to see things that are close up. Now I'll come back to the bird, but if you learned anything about accommodation, you've probably learned about accommodation in the human. So I'm going to switch to a monkey. And I'm going to ask anybody brave enough to tell me how accommodation works in the monkey. Oh, don't have it. Oh, here we go. This is what I made. This is actually a marmoset. So a tiny monkey. How does accommodation work in a human, which is the same in a monkey, in most monkeys?
Muscles tighten
Yeah,
all
so
around the lens.
so what kind of muscles do mammals have in their eye? Smooth muscle. Smooth muscle. So I'm gonna name 3 features of muscle, and you're gonna tell me which one is more advanced or more significant, skeletal muscle or smooth muscle. The first one is strength. Smooth or skeletal?
Skeletal.
Skeletal. Speed. Skeletal. And precision.
Skeletal.
Okay, so you've already learned almost everything you need to know about the difference between accommodation in mammals and birds. We suck. So this is a primate. Here's the ciliary body. And all of this pink area here is the ciliary muscle. I've already showed you the ciliary muscle from the bird, and you'd say, oh, well, this is much bigger than the ciliary muscle in the bird. But remember, this is skeletal, it's a smooth muscle, and the other one was skeletal muscle. So how does this muscle work? Let's draw a line, arbitrary line about here, and then we'll go to a little bit higher magnification. And we'll look at the smooth muscle fibers. If we go in front of that arbitrary line, the muscle fibers kind of center on, they would pull on an area here, and that is directly attached to the trabecular meshwork. This is Schlemm's canal, and that has to do with maintenance of pressure in the eye, drainage of fluid from the eye. So when this muscle contracts, when this smooth muscle contracts, it's bound, it's connected to the sclera, which is rigid, and then going inward from there, it's connected to itself. So you can think of it as being kind of connected to the sclera. When it contracts, it pulls this structure here and increases the resistance of fluid leaving the eye. If we go to the back, and we draw, here's our same arbitrary line. Now, if you look at where those muscle fibers go, they all kind of center in the back of the ciliary body, around here. So when that back muscle contracts, it's going to take this soft tissue of this is the pars plana of the ciliary body. It's continuous with the choroid. If we go further back, it's going to take that tissue, and let's go back to lower magnification. It's going to drag it forward. And if you drag that tissue forward a little bit, then it's going to narrow the aperture between the ciliary body here and the ciliary body on the other side of the lens. Ciliary body there and the ciliary body on the other side of the lens. And you can just barely see the lens is held in place by these little tiny fragments, threads. That are called zonular ligaments. So this gets now it gets totally counterintuitive. When the eye is at rest, in other words, when that ciliary muscle is not contracting, then the aperture here is wide. And what that means is that the zonular ligaments are under tension. So they're pulling outward on the equator of the lens. And what that means, we go to even lower magnification, is that the lens is allowed to be relatively flat. And if it's flat, then it's not focusing light. If the muscle contracts and the tension on the zonular ligaments is diminished, then the lens is more free to form its natural shape. So the elasticity of the lens, the innate elasticity of the lens, will cause it to rounden. And the only elastic structure in the lens is the lens capsule. So I'm going to attempt to show you the lens capsule in this marmoset. There it is. It's extremely small. So that the elasticity of the lens capsule will cause the lens to become more round, and that will then allow the marmoset to see things that are closer. Now, among mammals, primates have the best accommodation, and you can kind of see why. They live in trees. They have to grasp branches that are close to them. They hold things in their hand and study them up close. So they have adapted eyes that are, among mammals, the best for accommodation. And if you just think about that mechanism I just went over, it's piss poor. It's a lousy mechanism of accommodation. And for humans, as well as macaques, when humans get to be about the age of 40 or the equivalent in a macaque, you lose that altogether. You don't have any accommodation. Here's the only accommodation is you can take your reading glasses and put them on because you don't have any ability to accommodate any other way. Really lousy mechanism of accommodation. I'm going to put the marmoset away for the time being. Go back to our pelican. If you think about a bird, let's start out with a songbird, a little songbird. And the songbird is flying around. It wants to land on a branch. Here's my finger is a branch. It's blowing a little bit in the breeze. And that branch is going to be about that far from its eyes. And it's going to have to see that branch clearly in order to stick its feet on the appropriately on the branch. And it's going to move. The branch is moving, so it's going to have to change rapidly. Or now let's think of a woodpecker. The woodpecker is going up and down the trunk of a tree, and it's looking for insects in the bark, and that bark is about that far from its eyes, and it's using vision to find the insects in the eye. So it has a much greater need for more comprehensive accommodation than the mammal. Also, much more rapid accommodation, and like I said, 90% of the reason why that is because it has smooth skeletal muscle rather than smooth muscle. But let's look at how the two muscles work. Oh, here's the muscle. Looking on the wrong side of the eye. So here's Brooks muscle, the back of the eye. Notice these big nerves that are next to it. I'm not going to say that these nerves are entirely there to innervate the Brooks muscle, but you kind of get the impression that it's very heavily innervated and therefore pretty precise in its action. In Brooks muscle, and here's the sclera, and here's the ossicle. Brooks muscle. Just make sure I understand what I'm looking at here. Oh yeah. Brooks muscle is once again attached to the sclera, and now the sclera is attached to this bone. So this is even more rigid than the mammalian eye. So just like in the marmoset, when this muscle contracts, it grabs this soft tissue in the back of the ciliary body and the choroid, and it pulls it forward. Just like in the mammal, it decreases the aperture where the lens is held in place between the ciliary body on one side and the ciliary body on the other side. And if you look, you can see that the bird also has nonular ligaments, but I think we're allowed to forget about those because the main reason the lens is held in place is because of it has direct attachment of the plica, these branching segments of the ciliary body, direct attachment on the lens. And this pelican doesn't show that very well. So I'm going to quickly show you an ostrich eye. And I've chosen this ostrich eye because I know it has good, nope. That's a camel. Because it has, it illustrates that connection of the plica to the lens very well. First of all, the ostrich has a gigantic eye. When I get done showing this, I'll talk about eye size and ostrich eyes. So here is the Brooks muscle. Here is the pars pliccata, and now you can see real easily that those processes are directly attached onto the lens. If we go to the other side, the same thing. So forget about the lens capsule and the elasticity of the lens. Now we have a mechanically much more direct mechanism for changing the shape of the lens, i.e. a vise that squeezes the lens during contraction. And so it's much more direct. It's also under skeletal muscle control. So it's a much more direct mechanism. I'm going to switch back to the pelican now. Pelican. When I showed you the gross picture of the hawkeye, I pointed out this structure at the equator of the lens, which I call the annular pad of the lens. And so birds have this structure, annular pad, right where the pars pliccata attaches to the lens. So there's a difference in the way the lens epithelium is formed. And let's just contemplate that for a little bit. I'm going to attempt to bring out the, oh no. Yeah, I think now we can see that the contrast that these lens epithelial cells, as they approach the equator, get longer and longer. Let's go more towards the anterior pole of the lens. So just like in a mammal, the lens epithelial cells are continually evolving, and they undergo mitosis, and they're proliferating. They proliferate at an area more or less like this, and then they march towards the equator of the lens. So in the bird, as they're doing that march, you can see that they get longer and longer and longer and longer until they reach the equator, and then they switch and get shorter and shorter and shorter until they reach the back of this portion. Then they undergo a really complex change. They get short, they rotate, so they're now going in this direction rather than this direction. And then as soon as they get incorporated into the lens, then they become really long. Then they go all the way from the front of the lens to the back of the lens. And you can see the nuclei are maintained a short distance. Eventually the nuclei gets lost and we're into the what's it called? The center of the lens.
Nucleus?
The nucleus of the lens. Thank you. So that's a really complicated dance of formation that the lens undergoes. So, what is the structural and mechanical explanation for the annular pad? I have spent a lot of time searching the literature for someone who has attempted to analyze what the annular pad means. in terms of the function of the lens during focusing. So and I can't find it, so I just make it up. I think it must be somehow that having this structure means that the central portion of the lens, the nuclear portion of the lens, which is the optically important portion of the lens, is optimally adjusted during the accommodative process. So it maintains vision, in focus vision, both near and far, in a better way than it would be if you didn't have the annular pad. You're welcome to do your own searching on that. I haven't been able to find any better explanation than that. So that's Brooks' muscle. Let's go back and let's look at Crampton's muscle. And I think that there's a picture in the. We'll go back to the PowerPoint in a little bit after we do Crampton's muscle. So Crampton's muscle is the anterior ciliary muscle. And so it's here. Once again, in the back, it's connected to the sclera, which is connected to the bone, which is going to be most rigid. And then when that contracts, it pulls on this collagenous structure, which you can think of as sort of like a tendon. And that tendon, if we follow it forward, is continuous with the innermost and peripheralmost cornea. So if that Crampton's muscles contracts circumferentially, it's going to pull inward on the innermost and peripheralmost cornea, which is going to cause the peripheral cornea to kind of bulge inward. And that in turn is going to cause the axial cornea, which is where focusing happens, to increase its curvature. And I think there's a picture on here.
Accommodated by changing the cornea.
So they have corneal accommodation in addition to go back one. Yeah, so this is an attempt to illustrate the changes caused by Crampton's muscle on the cornea. It pulls the outermost, most peripheral cornea inward, and that'll increase the curvature of the central cornea. And if the corneal curvature is increased, then near vision is facilitated. Does that make sense? Much more, to a much greater degree than any mammal is able to accomplish. So I said the primates have the best of accommodation. There are some mammals like otters and other animals that hold things in their hand, like raccoons, also have enhanced accommodative ability, but nothing near what you'd expect to find in a bird. Okay, does that make sense? Okay, let's go back to the retina, and there's a picture in here about the retina.
I have no idea where.
Yeah, I have to go backward. That's accommodation in mammals again. Okay, yeah, here. So this, I have this section in my collection. I can show it, but it's nicely labeled here. And so this is does anybody here know what a plastic section is?
In acrylic?
I'm sorry?
It's in acrylic.
Acrylic, yes, the tissue, rather than being embedded in paraffin, it's embedded in an acrylic. And that means, among other things, that means that you can make a much, much thinner histologic section than you can in paraffin. We routinely embed our pathology specimens in paraffin. It means we can make about a 5 micron section, which is damn thin, but it's when you're looking at fine detail like we're attempting to look at here, it's everything gets blurred. So this is a plastic section. This happens to be a falcon retina, but there's really nothing special about it being a falcon. It's just a good example of a high detail avian retina. And so we're looking at the photoreceptors. Here's the nuclei of the photoreceptors, the outer nuclear layer. Here's a few ganglion cells up here. So we're not in an area of high visual acuity. It's just a nice section. It shows the population of photoreceptors in the bird retina. A lot of these things will be the same in a mammal. The inner segments of the photoreceptors contain what's called an ellipsoid towards the major part of the neural retina, and that's where all the mitochondria are. And then immediately next to the outer segment, there's what's called the paraboloid, and that is inhabited by rough endoplasmic reticulum, where amino acids are incorporated into proteins. But what I really want to show you here is this little round structure that's always just on the inside of the outer segment where light actually interacts with the tissue. And here's another one, here's another one, here's another one. And these are called oil droplets. And if you take a bird retina, this is a chicken retina. If you take a bird retina, open up the eye, remove the retina, and just put that retina on a microscope, flat mount that retina on a microscope without any other manipulations, and look at it through the microscope, this is what you see. Now, this has been color enhanced a little bit, so the color is not nearly as crisp as this if you do what I just told you. But these are the oil droplets once again. Notice there are 4 different shades of oil droplet in the falcon retina. I'm sorry, there's a chicken retina, and that's because the bird, unlike the mammal, has 4 different cone color variants. The bird is able to see in ultraviolet spectrum, short wavelength spectrum, in other words, blue, intermediate spectrum, we'll call that green, and then long wavelength spectrum, we'll call that red. Does anybody know how it works in mammals? I'm talking about color vision now.
Different options.
Different opsins, yeah. The proteins that regulate this are called opsins. So the primates, which have the best visual acuity among mammals, have 3. We have a short wavelength, which is blue, a medium wavelength, we'll call it green, and a long wavelength, which we'll call red. But only 3. All other mammals, besides primates, have at most 2. And they have blue. Not every mammal has blue, but most mammals have blue. And then they have a medium wavelength. And again, if you read about this in books, you get different opinions. Some say that most mammals have then a green photoreceptor. Some say then that most mammals have a red photoreceptor. And I have no way of knowing. I tend to think that most mammals are missing the red photoreceptor, but I may be wrong about that. So birds have enhanced color vision for 2 reasons. One is that they have 4 different color variant, color specialized cones, UV, blue, green, and red. And the other is that the light, before the light gets to the part of the cone that actually responds to the light, it has to go through a filter, and that filter is specific. These red dots would be for a red cone. The yellow dots would be for, I don't know, yellow. Let's just call it yellow. Here's one that's sort of green. And the clear ones here are for UV light. And the clear ones function as a UV filter to prevent potentially destructive UV light from reaching the outer segments of the light. So you'd think then that birds, that's another reason why birds have much more sophisticated vision. Well, think again, because virtually all vertebrates, going all the way down to very primitive fish, have oil droplets. Birds have 4 different color variants of oil droplets, so they are specialized. Turtles, I think, have 3, or at least some turtles have 3. Other ones, vertebrates might not have colored oil droplets at all. But almost all vertebrates have oil droplets, except again, mammals and snakes. And stick a pin in that mammals, because I'm going to show you the exception to the rule among the mammals. Another thing to notice is that some of these photoreceptors are double. This would be called a double cone. Here's another double cone. And I haven't yet found someone that's willing to say what the function of a double cone is, but they exist again in virtually all vertebrates, from fish. I don't know about sharks, to up to mammals. Mammals don't have double cones, except, stick a pin in it, because there is an exception among the mammals that where there are double cones. Okay, one more thing with this slide. The good opportunity to look. This is a rod. This is a rod. This is a rod. Here's a cone. 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 Notice that the rod outer segments, where the light interacts with the protein, is very long, and the cone outer segment, where the light interacts with the protein, is very short. Can anybody offer an explanation for why that would be? What's the function of the rod? Dim light. It wants to function in dim light. Let's say a dark night where there's a crescent moon and starlight available, so that only a very small amount of photons are going into the eye at any one time. We used to have a Chesapeake Bay Retriever, and I would let her out at night, and I'd go out in the stoop with her, and to me, everything in the yard would be dark. She would stand on the stoop for about 20 seconds, and then she'd dash off after something. Lord knows what it was. She apparently knew what it was. I was not able to see anything, but she was able to adapt to dim light. We're going to pick up on the topic of visual acuity versus visual sensitivity in a little bit, but the rod is a specialist in visual sensitivity. The cone is a specialist in visual acuity. So, having said that, what's the advantage of a rod to having a long outer segment?
The distance the photon has to interact.
That's exactly right. It means that let's say he's dealing with starlight, and this rod is getting 2 photons. That photon is probably going to be absorbed somewhere along this long segment here, and once that photon is absorbed, it's going to stimulate some kind of electrical activity, and my Chesapeake Bay Retriever is going to recognize that as a stimulus and take off in the yard after. I always assumed it was rabbits, but could be anything else. And so it's the advantage of the rod to have a very long outer segment. Why then is it an advantage to the cone to have a very short outer segment? The cone is functioning when there's plenty of light in the daylight. Light, the amount of light available to the cone is unlimited. It doesn't have to worry about that. What it has to worry about is making the correct decision. If they've got red light, it's going to respond appropriately within the retina to signal a red light. If it's a green light, it's going to respond appropriately in the retina. So it doesn't want to make a mistake. So it doesn't want this long opportunity for light to pass through, and then all the red light, let's say this is a red cone, all the red light would be absorbed here, and the light that would go to the end here would include some green light or UV light or something like that. And so that's going to stimulate a response as well. So the cone is likely to make some mistakes. The cone wants a limited outer segment to eliminate mistakes.
I have a question.
Yeah.
So I could picture how wavelengths of light would be coming, or a beam of light or a photo, whatever you want to call it, coming at an angle.
Uh-huh.
That's not gonna go through the oil droplet. How do you explain that? Is it because the RPE is shielding all the way around that oil droplet, or?
So that there's an answer to that, and it's complicated optics that I'm not going to attempt to explain. Yes, the photoreceptors are largely at an angle, and I believe that that angle is adjusted for how the light comes in, and the light when it's in the photoreceptor acts like a fiber optics tube, so
Okay.
that it gets directed appropriately into the cell. Correct me if I'm. There are probably retinal function people who know that better than I. And I'm prone, I'll warn you ahead of time, I'm prone, if I don't know something, to make us to attempt to try and use logic, which is you're always on thin ice when you're using logic to.
So wait, well, my question is, so like, I mean, I understand light coming directly in this,
Yeah.
but what about a light that's coming here? You know, or an angle that's coming right down here?
I think the fiber optic explanation is pretty good. I'm not a photoreceptor person,
Okay,
but if you shine a flashlight on a fiber optic cable, you're not screwing up the signal inside the fiber optic cable, right? So
Right.
it's the same kind of idea, I think. If the light comes in from the lens at the right angle into the photoreceptor, it kind of tunnels into the crystalline structure of the photoreceptor in that outer segment. And if you get light coming in at an angle from the side, it just doesn't make, it either gets reflected off, or I don't know.
Yeah, the physics of that has been worked out pretty carefully, and it goes over my head, so I usually don't try to explain that.
It also just shouldn't be nearly as much light coming in at different angles. Like the back, if the optics are right, the vast majority of the light should be coming in from the pupil
Okay.
down.
Yeah. Okay. I should also say that the light, as it passes through the retina, travels through its cellular cytoplasm, sort of like a fiber optics cable as well. So it's delivered directly to the outside of the lens. The retina is transparent. I don't know if it's transparent to the extent that carefully constructed glass is transparent, or if it's just more or less transparent. When we see the formalin fixed retina, it's no longer transparent, it's translucent.
Yeah, it can't be that great because when you do tissue clearing, you can make it clearer. And when you do adaptive optics, you can improve the resolution of imaging the retina from the outside. So there's always interference in the path.
Okay, I'm going to show the eye, just like we showed the pelican. I'm going to show the eye of a golden eagle. We'll talk about some aspects of visual acuity. There's some ways in which this golden eagle shows features that are different than the pelican, but I'm not going to dwell on it because for the most part it's the same. When you're talking about bird eyes are so well adapted for visual acuity that there's not that much anatomical difference between one bird eye and another. Bird eye is a bird eye. You got big ones and little ones. So again, same thing. Here's the scleral cartilage. Is that in focus?
yes.
Here are the muscles. Here's the annular pad. But we're going to look at the retina. Here's it's a better section of the pecten oculi. You can see this nice shading of the photoreceptors. To some extent, you can see the choriocapillaris a little bit better than you could in the pelican. So this, look at the number of ganglion cells. Again, if you're looking at retina histologically and you want to know, are you looking at an area of the retina that's adapted for visual acuity? Look for the number of ganglion cells. The more ganglion cells there are, the more likely this is an area of the retina that's adapted for visual acuity. And so we're going to be very close to a structure called the fovea, although we're not in the fovea. But I think you can get the impression that the information going to the brain is going to come through the ganglion cells. Also notice how thick the inner nuclear layer is. And I know almost nothing about this, but I'm told that compared to the mammal, that neural processing occurs to a much greater extent in the retina as opposed to the brain, so that the image that goes to the brain is already somewhat neural processed. And I don't have any better words than that to describe it. The other feature to look for with regard to where you are close to a fovea is to look at this innermost layer. This is the nerve fiber layer. The nerve fiber layer contains the axons of the ganglion cells, and the nerve fiber layer emanates from the fovea. It does not pass over the fovea. So the nerve fiber layer, when you get into the fovea, disappears. And now we're very close to the fovea, so you can see that it's disappearing. Let's keep your eye on that nerve fiber layer as we go towards the optic nerve, and you can see that now you're getting axons of the ganglion cells getting thicker and thicker and thicker and thicker until you reach the optic nerve. And now all the axons are coming here, and then they'll turn into the optic nerve and go to the brain. And I think that's all I really want to show in this golden eagle. So I think it'd be a good time now to take a bathroom break, about 10 minutes, and come back, and we'll pick up from there.
Thank you. Put that back, I have a question.
I have a question from my wife. That's gonna be, yeah, that can't be good.
Where's my water bottle?
You want to go to the same spot?
Yes.
What's the question, young lady?
So I see the pigmented layer. And I
Yep.
see a whole bunch of. Yeah. Which layer are the ganglia? Are they? Like
The
going-
the innermost cellular layer is the ganglion cells. So there they are.
down there? Down there? Those kind of larger cells.
Those are the nuclei of the ganglion cells.
Right. And then what's that whole thing that looks like an elastic band?
This?
No,
This,
that,
that's so
Oh.
cool. It's
Yeah.
like laminated.
Yeah.
That's the the inner plexiform layer. That's where the ganglion cells are talking to the inner nuclear second order neurons. That's incredible how well detailed it is there.
so those are separate cells from the ganglia.
No, this
Well,
is all like.
the
cellular
dendrites,
processes.
the dendrites of the ganglion cell would go down here and intermingle with the axons of the outer nuclear layer. So this is axons and dendrites and synapses.
I've never seen that before.
Really thick.
I'll have to get you a golden eagle eye.
Thank you.
I kind of have a question about the inner segment and outer segment.
Yeah.
So I've worked with dogs that had subretinal injections in the
Mhm.
past, and after the blebs reattach, I've noticed that sometimes within the bleb area you have this darkening. And I remember my mentor saying that, oh, maybe the inner segment, outer segments are like off angle, and that's why the light reflects a little bit differently.
And who's your mentor?
William Beltran.
Oh, okay, yeah.
I wonder, like, does that mean that?
I am not going to attempt to say anything different than William Beltran about the photoreceptors.
they fix, did they like right?
For the most part, they will function again. I did a study once with a fellow here, retinal specialist called Mike Nork, where we use ultrastructure to study the difference, the return to normal of the bleb.
How-
And we did discover something, but that was a number of years ago. I can't remember. It was pretty subtle. But I don't remember what it was.
Just goes 100% back to normal once everything.
They function normally.
How it just figures it out.
Yeah.
So Jennifer, are you still working with Professor Beltran?
Actually, I am.
Oh, okay.
I'm still finishing up some of the research stuff that I have to publish.
So
I've got one more paper left.
Beltran is in at Penn. He works with Gus Aguirre.
Oh, we love Casaguiri.
It's so nice.
Yeah. Among other things, colorful.
Yeah.
Yeah.
I know.
Thank you, Dr. Shaw. Turn down the controls.
Yeah. I recently, when we were doing the Mary review last
Yeah.
week.
Yeah,
Um,
I'm sorry, I was not here.
Oh, yeah, somebody piped up and said, oh, falcons don't have a blind spot. Is that true?
No.
That's what I thought. I was like, no, there has to be a blind spot.
Yeah.
There has to be a blind spot.
If you have an optic
Okay.
nerve.
That's what I thought too. I was like, and he was adamant.
If you don't have an optic nerve, then everything's blind.
Right.
Octopus doesn't have blind spot. Maybe he was mistaking falcons for octopus.
Sure.
So put this one on.
I was just talking to our department.
You what?
We have a bit of a lull. There are some species that kind of have yellowish lenses, lens sections.
Oh, yeah.
And I know you were studying like flavonoid rich, like eyes and things like that at one point, like the lemurs are at least kind of looking at them.
Mhm.
Like, do we have a specific explanation for why some?
Yeah, if you come across an eye that has a yellow lens, it's likely to be an animal that spends its time in bright light. So primates, humans have a yellow lens, horses have a yellow lens.
Ground squirrels.
Ground squirrels have a very yellow lens.
Squirrels.
What we just came across one, the meerkat has a yellow lens.
We have a yellow lens.
Yes.
I learned two things today. Among other things.
And the function of that is ultraviolet light protection. Some people still in the bathroom.
Yeah.
I can't see.
Hi Doris, how's it going?
Gee.
Oh, no.
So is this Marcella? I want to say hello to Marcella. Hi Marcella, I haven't met Doris. I don't think we've met. Nice to meet you.
Okay, so sticking with birds, and we're going to talk about visual acuity.
I don't think it's been quite 10 minutes.
Oh, okay.
Started.
I can't.
3:29, okay. We got 4 more minutes.
Okay. I'm antsy to go.
Yeah.
Put it back in that.
There was back to the conversation about post-retinal like detachment, essentially like the attaching not quite the same way. There was a Mary discussion at one point, like one of those Friday rounds where like a retinal detachment surgeon was talking about how that is things, but then like the like shifting relative where the blood vessels were initially.
Mm.
Things. Funny. If we could find the recording of that, I could send it to you.
Yeah, that's kind of neat. Yeah, if you're a mammal that does have blood vessels in the retina, then the reason why you don't see blood vessels all the time is because your brain adjusts for that and erases the effect of the blood vessels.
Science, let's see.
But if all of a sudden the blood vessels go to a different part of the retina than where they used to be, then you can see where the image of where they used to be. Or if someone takes a bright light, like a slit lamp, and shines it in at an angle, then you can see a shadow of the, yeah. You can actually see the avascular zone of the fovea.
Yeah, like basically that discussion too was, like retinal reattachment doesn't restore like a good amount of function, but even like those subtle shifts can cause like, you know, reasonable food, like aberrations, I guess, in the human patient. We can't ask the dog, of course. But,
Okay, so am I allowed to go?
Sure. Oh, wait, hang on. Yeah, go ahead, go ahead. Wait, hang on. Oh, hang on.
Just a
Hold on.
second, just a second.
Doris wants to take a picture.
I'm gonna take a picture. Is there anyone who objects to having me take a picture of them? Okay, we need pictures of people at events for the McPherson ERI Research Institute Outreach Committee promotion.
Okay.
I can't do it of the kids.
While Doris is taking her photo, somebody asked a question in the chat here. Is it yellow only after it fixes in formalin?
After it fixes in formalin is the only time I ever get to see the lens. I don't think so. Does anybody here work with
Unfixed.
ocular media and mice have what?
Mice have
Clear lenses.
Those are definitely clear.
But those
Yeah.
aren't the species that we listed as having yellow ones, so.
Which while you were still out, so it's a species that have high light exposure and so like.
So mice.
So another protective mechanism against ultraviolet light is the macula lutea, the yellow color of the inner retina over the retina. And primates have a macula lutea. I don't know if they all do, but humans and gorillas do. Chameleons have a macula lutea, which I have pictures of. And I don't know who else has. Okay, visual acuity. The portion of the retina that is most useful for visual acuity in other words, it has the most ganglion cells and the highest number of cone photoreceptors is going to be the fovea. And the fovea is defined by this V-shaped indentation. And then I already mentioned these are the shadow of the ganglion cells, and then the black here is the shadow of the nerve fiber layer. So the nerve fiber layer disappears over the fovea because it emanates from the fovea. It doesn't cover the fovea. Then it gets big again, and then many birds, let's just say some birds, including all of the diurnal, all of the daytime raptors, eagles, hawks, falcons, etc., have not one but two fovea in each eye. So this is a Peter Sukup and Simon Pot did a study on that, and they got these wonderful sections in which they sampled both fovea at the same time. And so this is just a good example of a double fovea in a kestrel in this particular example. Among mammals, the only mammals that have a fovea are primates. And most of the old world primates have a fovea. Most of the new world primates have a fovea. But if you get into more primitive primates or nocturnal primates, then they no longer have a fovea. A lot of mammals that don't have a fovea will still have a portion of their retina that's adapted for visual acuity and it'll be called the area centralis. And although there's not a pit, as you find in the fovea, there will be an increase in number of ganglion cells and a diminution of the nerve fiber layer in the area centralis. And I don't think I have an example histology slide that shows that. Next slide. So people try to answer the question, well, what do the nocturnal, what do the diurnal raptors use the two foveas for? And you can see that the temporal fovea, the fovea on the outer portion of the retina, when the bird is looking at the image is in focus on both fovea at the same time. So that means that in the temporal fovea, it provides stereopsis for depth perception. The central fovea is unique to the eye. So you'll see birds sometimes sort of eyeballing something, it'll stick its eye up and look directly at it. When it's doing that, it's using its central fovea and seeing with just one eye. Okay. Does that make sense? We're going to jump back temporarily to
So do owls have two fovea?
owls are nocturnal, they're not diurnal. It's variable. Some owls, there are diurnal owls, but I think none of them have two fovea. But many owls, and I don't know about all of them, the great horned owl, for instance, has a fovea. It's one of the very few nocturnal animals that has a fovea.
Yeah, I was just thinking about the eyes on the front of their face versus on the side.
Yeah. So I talked about the shading of the photoreceptors, and I said it goes way back to the fish. In fact, this phenomenon of shading of the photoreceptors is most prominently seen in the fish. And this is I have to remind myself, this is a zebrafish. Oh. So this is the head of a zebrafish. And we're just going to look at this phenomenon of shading of the photoreceptors. In a fish, in many fish, it's always skeptical to say all fish, the shading of the photoreceptors is dynamic. So that if this fish dies in the bright light, as this zebrafish did, then the pigment is going to move forward to shade the outer segments of the photoreceptors. If this fish died in the dim light, in the dark, the pigment will move backwards to expose the photoreceptors. We're going to talk about underwater vision later, but I'll just prime the pump here by take a look at the lens and the vision, of course. Can you focus that? There, this is better. Pretty good. Vision, of course, was entirely underwater for the first 200 million years of vertebrate evolution. So there was no reason to have anything other than this. And the exquisitely round and dense lens is an adaptation for vision underwater. We're going to come back to that when we talk about whales and dolphins and seals and what have you. And then I already said that if you're going to find blood vessels in the retina, that it's going to be a mammal. Well, this fish has blood vessels in front of the retina, actually in the vitreous. And this structure is called the membrana vasculosa, but it's not in the retina, it's in the vitreous stretched over the front. So if you do fundoscopy on a fish, it's going to look like there are blood vessels in the retina, but they're actually in front of the retina and not in the retina proper. That's true in some snakes, and maybe in some other reptiles. I'm not sure. Amphibians, for sure. Okay, remember that round lens, because we're going to come back to that when we talk about oh. I already showed you a picture of the double fovea. I have our more primitive attempt to capture the double fovea in a slide here, but it doesn't do it the justice that the section that Dr. Sokup and Pot did. Okay. You should be left now with the impression that birds have eyes that are exquisitely adapted for visual acuity, right? And we've already gone over that birds basically are reptiles. So we're going to look now at a Komodo dragon and a iguana. This is the Komodo dragon. Oh yeah. So this is the low magnification of the Komodo dragon. And you'll notice that there's a cartilage, there's a scleral ossicle. Oh, we'll go to higher magnification, but there's an annular pad. I've already told you that all reptiles and only reptiles have skeletal muscle in their iris and ciliary body. So there's skeletal muscle there. Notice that it doesn't have a pars pliccata. The ciliary body does not touch directly on the equator of the lens. And although the skeletal muscle in the ciliary body, the orientation of that muscle is different than the birds, and I've never really been able to figure out exactly how it works in each type of reptile. But I would be willing to bet that accommodative mechanisms are the same, that this animal accommodates by direct pressure on the lens, decreasing the aperture of the space between the ciliary body and the ciliary body. So a lot of the features that we talked about that seem exotic and sophisticated in the bird eye are already present in the Komodo dragon, and I'll show you that they're already present in the iguana. Iguanas and the Komodo dragon are quite distantly related to birds, although they're all reptiles. If you remember back to that phylogeny, the crocodilians and the turtles are more closely related to birds, but the structure of the eye is considerably different in those. Crocodilians, for most of their existence, are pretty exquisitely nocturnal, and the evolution of turtles remains an enigma. I don't know that much. I don't know if anybody really knows that much about them. Okay, so here we have going the back of the eye. We're looking on the inner portion of the retina, and here's this structure that once again is highly vascular. I'm going to say it's analogous to the pecten oculi, but it's not a pecten oculi. It's entirely conical, and this structure is called the conus papillaris. So one can say, well, the conus papillaris transitioned into a pecten oculi, and that may be true. We talked about the tuatara earlier, but the tuatara doesn't have a vascular structure in the back of the eye. So I think it's more likely that the conus papillaris and the pecten oculi are evolved in the different species independently. I don't know about that. I don't know that anybody's really studied that. We don't have the ocular structure for extinct animals. So it may be hard to designate that. But this is the conus papillaris, which is a feature of the lizard forms. So iguanas, chameleons, geckos, dragons, and there's probably some other ones too. They have oil droplets. I'm not going to be able to demonstrate the oil drops, but they would have oil droplets. They would have double cones. This is an iguana, same thing. Here's the conus papillaris that's kind of pumped over on itself. And now you can see here, there's a little bit of a shallow fovea. And the Komodo dragon probably has a fovea as well. So specialists in visual acuity, not just birds, but the ancestors from which birds derived are specialized for visual acuity. So one might say then that birds and reptiles have better eyes than mammals. So we're going to talk about the difference between visual acuity and visual sensitivity. I already described my ability of my Chesapeake Bay Retriever to see things that I can't see in dim light. And so this is kind of a standard generic ocular morphology for a mammal. This is a
Step back to the pecten oculi and the other conus one.
yeah.
You know, as a physiologist.
Yep.
I'm curious about just how effective those are at oxygenating the vitreous, and is the vitreous different in these species so that, because, you know, I think of vitreous as just like jello, and
Yeah.
moving oxygen through jello is challenging.
Yeah. So
You know, how well do they work? I mean, it's fortunate that there's a blood supply for the rods and cones.
yeah.
And it's good that, you know, the outer or the inner retina doesn't need it as much, but how much does it get?
So if you were to tell me that that's impossible for nutrients to transfer to the inner retina in that way, I wouldn't be too disturbed by it, because there's another function for the pecten oculi and the conus papillaris, and that's to contribute to aqueous formation.
Ah,
Do you know what Cloquet's canal is? Cloquet's canal is a clear area in the right dead center in the vitreous that goes to the back of the lens.
Okay.
And so aqueous travels from the pecten or the conus through Cloquet's canal and contributes to aqueous formation. So maybe that's the whole reason for why they have them, but most people who discuss the function of the pecten and the conus papillaris mention nutrients to the inner retina.
just wonder if anybody's ever actually measured the oxygen tension in the vitreous of these animals.
Yeah, I really don't know. I know that in humans, well, I don't know. I have vague memories of talks in which they talked about that. So this is a tiger. Well, let's go down here. And I'm using it as a sort of a generic.
Here's a stand for it.
Oh, yeah. I'm using it as kind of a generic mammalian eye. The reason why this histosection of the tiger eye is yellow is because it was fixed in Boone solution. So this is remnant picric acid that didn't wash out of the tissue, not because it's got a yellow lens. So a couple of things to pay attention to. One is that the cornea, the relative, if you look at the circumference of the eye and you ask yourself the question, how much of the circumference of the eye is devoted to the width of the cornea? Generally speaking, in mammals, there's going to be more circumference of the eye that's devoted to the cornea. I like to call that a picture window cornea to let more light in. The lens is as a general rule is more round and more positioned towards the middle of the eye. The tiger gets away with a relatively flat lens forward in the eye because they have such a big eye. Which the larger the eye, the better visual acuity you're going to have, or the better your vision is going to be in any dimension. Hence, although birds can be pretty small, they go way out of their way to devote space in their head, in their little tiny heads, to having as big an eye as they could possibly manage. Not so much in mammals, although if you're going to find a really big eye, like in a horse or in a giraffe, it's usually going to have a very big head as well. So tigers do very well hunting at night. They're very adept at hunting in total darkness, and they also do quite well hunting in the daytime. So they're crepuscular. They're out at various times, various light intensities. Now let's look at the retina. What you're going to find in a mammal compared to a bird is a very thick outer nuclear layer. Remember, these are the nuclei of the rods and cones, and the mammals are going to be light sensitive, adapted. They're going to have a larger number of rods compared to reptiles. And rods, if you remember back to that plastic section of the falcon, rods take up less space in the retina than cones. So you can cram more of them in, and each one of those rods has to have a nucleus. So there's going to be more nuclei in the outer nuclear layer. And then there's going to be fewer ganglion cells because visual acuity is not the goal here. Visual sensitivity is the goal. So here's the strategy of the nocturnal retina. You want to have a whole segment of photoreceptors, a whole segment of outer segments functioning together. So if some small amount of light comes in, some photoreceptors in this whole segment are going to interact with that light. And then they're going to, that whole bunch of cells is going to hone in on a small number of ganglion cells. And so that'll allow those ganglion cells to register some kind of impulse to be sent to the brain. Probably not visual acuity, probably not reading, probably not studying close up. Maybe, perhaps, if someone knows better than I, I'd love to know what it is that nocturnal animals see. I'm going to guess motion or coarse figures, but that's going to be the goal here is visual sensitivity as opposed to visual acuity. Does that make sense? And the tiger, remember, is sort of in between. It's emblematic for the mammalian eye. It's not the nocturnal extreme. Does anybody know what this structure is in back of the retina? That's the tapetum lucidum. Does everybody know what the tapetum lucidum does? Usually we just call it the tapetum. It reflects the light back. It's the reason why if you shine a flashlight at a dog or a raccoon or a possum, at night, that it flows, it shines back. It's called eye shine. And once again, the reason, and that's a feature of eyes that are adapted for visual sensitivity. And the reason is, notice that the retinal pigment epithelial cells in this area are not pigmented at all. And that's because they want that reflected light to go back through the retina again, and have another chance to register as it passes through the retina going the other direction. I told myself, well, that doesn't make sense, because no way is this reflective. It's not like a mirror. It's not going to go through the same photoreceptor as it went through before. And I was talking with Dan Erik Nilsson, a vision specialist from Sweden, and I told him about my worry about that, and he said, "Oh no, you're thinking of it all wrong. It doesn't have to go through the same photoreceptor. It only has to go through the same ganglion cells, which is what's sending the signal to the brain." So that explained everything. It can take this, it can pass through anywhere in this area that's feeding into the same ganglion cell, and still be useful for constructing visual sensitivity. So any questions about the tiger? We're gonna come back and talk about the tapetum, hopefully before we leave today. Okay, I'm going to show you two eyes from mammals that are nocturnal specialists. The first one is the leopard cat. Now, this isn't the leopard. This is a cat called the leopard cat. It's also called the Fisher cat, which is a distinctly nocturnal variety of cat, wild cat. Didn't break. So here's the Fisher cat. Now, we're going to pay attention to the cornea, and this has got this wraparound cornea. So a large portion of the circumference of the eye is devoted to the cornea. I call it a picture window cornea. The purpose is, I think, obvious. It's to let as much light as in as possible. If you're going to do nocturnal photography, you want a great big wide lens to let in as much light as possible. And then you want to have a lens which is going to take the little bit of light that's available, and it's going to center that light. It's going to focus that light on a small area of the retina, so that whatever light, whatever photons that are available, they'll be bunched up in the retina due to the focusing mechanism, so that some sort of image can be interpreted by the retina. So they tend to have a round lens with a lot of a lot of refractive power, and the lens is going to be positioned more towards the center of the eye. And then we'll look at the retina. So even more thick outer nuclear layer and relatively few numbers of ganglion cells. Here's the tapetum again, and both of these are Felidae. Both of these are cat like mammals, so they have a very similar kind of tapetum, and there are different structures of different tapeti. I don't know if that's correct or not. But this is archetypical of a nocturnal retina, and using the thickness of the different layers is quite useful in mammals. It gets a little bit less useful in reptiles, and we'll talk about that again when we come to geckos. I think I want you to go to We'll, after this, we'll go to that. Go to this, and then the very last one. Well, the very last one. So this is from that book that I showed you, Wall's, the vertebrate eye and its adaptive radiations, and it's an illustration of the retina from a nocturnal animal and a retina from a diurnal animal. And it's showing oil droplets. So this is probably from a reptile. This schematic is not quite as useful in reptiles as it is in mammals. But again, you can see how thick how many rods there are compared to cones, how many nuclei there are in the outer nuclear layer, and then relatively few ganglion cells, whereas in the diurnal animal, lots of ganglion cells, relatively few cells in the outer nuclear layer, and then more cones than rods. And go to the next one. So these are the squirrels are nice for this. Most squirrels are very diurnal. They're out in during the daytime, and that's particularly true of the ground squirrels. I'll show you some ground squirrels slides in a little bit, but this works out very nicely. The exception among the squirrels is the flying squirrel, which is an exquisitely nocturnal squirrel. So here you have your ground squirrel, lots of ganglion cells, very thin outer nuclear layer, and then basically lots and lots of cones. For a long time, it was thought that the ground squirrel had a pure cone retina, but that's not true. Is anybody here studying ground squirrels? You know, they're studying in Oshkosh in particular. They have a colony of ground squirrels, but lots and lots of cones, relatively few rods. Whereas the nocturnal flying squirrel, relatively few ganglion cells, lots and lots of photoreceptors, very long outer outer segments, and so typical of a nocturnal eye. Remember how I told you that mammals don't shade their photoreceptors? Well, here's an exception. Here's one exception to that rule, and that's the ground squirrel. The ground squirrel comes out in the daytime. They live in burrows. They come out in the daytime to graze in the prairie, and they designate one ground squirrel to be a sentinel. And the sentinel ground squirrel's job is to look for predation. And the main place it's going to look for predation is for hawks. And hawks are likely to predate squirrels by diving right out of the sun. So these guys are adapted for being able to recognize something coming right out of the sun. So among all mammals, they have adapted some shading of the photoreceptors. They also have a very yellow lens to protect for UV light.
They could use the 13-line ground squirrels, the same ones that.
Yeah, this is the 13-line ground squirrel. We used to have a researcher who studied not retinal function, but hibernation. And so she provided us with 13-line ground squirrels. Exactly.
I think they're gonna actually move the colony to the medical college because Dana's retired. Oshkosh is pulling the plug on just about everything, and the 13-line ground squirrel is a really valuable model of a diurnal animal. Much better model actually than a rat for visual studies.
Yes, so MCW does have the ground squirrels, and they just made albino model.
Huh.
So they're getting the genetic engineering is working on them.
Interesting.
There's a big focus at NIH on these because of a lot of the hibernation changes. I mean, everything dies back in the eye, and yet it all comes back again.
Well, not everything. I have electron microscopic images of the hibernating ground, and there's rather subtle changes that happen in the outer segments of the photoreceptors.
Oh, it's fascinating.
It is.
So they were able to actually do the entire genetic sequence of these animals, which means they're really a valuable tool now.
So whenever I talk to a ground squirrel scientist, talk with vision, I suggest that they do an experiment to find out what if you're exposed to brighter and brighter light, there must be some amount of bright light in which they can no longer see. And my hypothesis is that that is a brighter light in the ground squirrel than it is in other mammals. But I don't think anybody's ever done that experiment. I have talked to Dana several times.
Dana is fighting her bones now, or.
Okay. Okay, well, she's excused.
Yeah, no, she's had it. She's enjoyed it, but yeah,
She's a favorite of the Coppola lab.
she's amazing. She's a wonderful person.
So this is an African rodent called I have to look at the back. Springhouse. Yeah, springhouse, which is a very nocturnal rodent. So we're looking at the same things. Picture window cornea, roundish lens that's toward the center of the eye. And then we'll look at the retina. And again, lots of nuclei in the outer nuclear layer, relatively few ganglion cells. And then here, the springhouse is one of the very, very few rodents that has a tapetum. But in this case, it's called a fibrous tapetum. So the reflective element of the tapetum is collagen. It's actually iridescent, so it's not just a matter of reflection. It's a matter of establishing iridescence. We just discovered, we didn't discover it, but we received an eye from a lowland paca. Is that right, Megan? A lowland paca, which also has a tapetum. So, but unfortunately, that tapetum has already been studied, and it's a cellular tapetum, unlike this one. And looks to me like it's very primitive in terms of how it's forming in the eye, which I think would be an interesting thing to study. So this is another nocturnal specialist mammal.
So does the tissue keep re-evolving from a different source every time, or is it?
Yeah, I believe so. The tapetum.
Yeah.
Yeah. So many of the animals that we deal with as veterinarians have a tapetum. Those related to horses, those related to cows and deer and what have you. Those all have a fibrous tapetum. And then the carnivora, dogs, cats, skunks, raccoons, weasels, have what's called a cellular tapetum. And I showed you the cellular tapetum from the cat varieties. But then there's a dog variety as well. The cat variety tapetum, the reflective part of the tapetum, which again is iridescence, comes from reflection off the structures in the cytoplasm of the cells. And in the cat, it's a combination of protein and riboflavin. In the dog tapetum, and so it's the anything you'd recognize as a cat. But in addition to that, hyenas are categorized with the cats. The dog type tapetum, which is most of the rest of the carnivora, bears, otters, sea lions, etc. The reflective element is a combination of protein and zinc. And then meerkats and who fights with cobras? Mongoose and mongoose are carnivora, but they're exquisitely diurnal, so they don't have a tapetum.
Hmm.
Also, camels and llamas are exquisitely diurnal hoof stock, and they don't have a tapetum.
Yeah, because I was doing, so I do zebrafish research. But I have a collaboration where we're doing spotted gar, and the gar has one.
Yeah.
Oh.
Zebrafish don't.
Many of those fish have that's what's called a retinal tapetum, and the reflective elements are in the cytoplasm of the retinal pigment epithelium. And oftentimes, I'm not going to use the word always, but oftentimes it's the amino acid guanine that's reflective. Among mammals, opossums have a white tapetum, and their tapetum is in the retinal pigment epithelial cells, and it's made up of little droplets of oil. So it's not iridescence, it's just a white surface. And the same with at least some fruit bats have a white tapetum.
Both dorsally and ventrally, and an RP that was pigmented throughout.
Okay, so we're going to start talking about the tapetum. That's where we're going right now.
Can anyone name an 8-legged creature that has a tapetum? What kind of spider? Probably lots of them, actually, but yeah, wolf spiders. Possibly. I don't know. I just know wolf spiders for sure.
When I was a kid, I would go out at night hunting for
Yeah.
spiders. with
Yeah, exactly.
Yeah.
Flashlight.
A headlight, yeah.
I've never been able to do that. I think I'm always too lazy when it gets dark. Okay, so we talked about ground squirrels. I think I have ground squirrels in here. We're talking about that separately. We may come back to ground squirrels later. So this, I'm told, is the mammal that has the purest cone retina. Remember, I told you that it used to be thought that ground squirrels had a pure cone retina, but this mammal doesn't have a pure cone retina, but has at least some people are led to believe that it has the purest cone retina. And I'm going to show you because it's an interesting looking retina, and I'm not going to be able to tell you much about the animal.
Wow.
Look at all the ganglion cells, and look at how thin the outer nuclear layer is.
Wow. This is a mammal, you said?
This is a mammal. And this is a tree shrew. The tree the tree shrew is a enigma. Maybe it's less of an enigma now than it was a few years ago, but there was a time when it was thought to be the most primitive of the primates, but I think that's no longer what people say about the tree shrew. And I wasn't going to show it here, but Janice said she was interested in the function of the photoreceptors, so I said, "Oh, I got to show the picture of a tree shrew." And I really don't have much more to say about it. It's a diurnal animal, not surprisingly.
Probably a lot less fun to work with than the 13-line ground squirrels.
There are some, I don't remember, there's some really interesting reasons why people study tree shrews. It's behavioral, and I've forgotten what it is, but it's totally unrelated to vision, something embryologically, hormonally related, I think. Anyway, that's a tree shrew. That's especially for Janice. Let's just talk about the tapetum. I showed you an example of a fibrous tapetum and a cellular tapetum, and they occasionally pop up in primates. Your most primitive primates like lemurs and prosimians, tarsiers, and what have you, have a ribosome tapetum. If you get a lemur or a bush baby eye in formalin, the formalin is often bright yellow, and the reason for that is the riboflavin leaches out of the tapetum and causes everything to be yellow. But those are the only primates that have a tapetum. We talked about a couple of rodents that have a tapetum. There are a few marsupials, the opossums, the koalas have a tapetum, and I don't know if I can think offhand of another. Opossums have a retinal tapetum, and I talked about it. I think I have an opossum in here, and koalas have a fibrous tapetum. So someone asked, is this a structure that evolves over and over again in different forms? And the answer to that question, quite obviously, is yes. But if you get into your most iconic mammals, either your hoofstock or your marsupial or your carnivora, then tapetums are the rule rather than the exception. And they're broken into a fibrous tapetum, which is typical of the hoofstock, and the cellular tapetum. And then you have a particular kind of tapetum in a feliform, and another kind of a tapetum. I don't think they're called the caniform, but all the other carnivores that are not feliform. So where I'll ask our veterinary ophthalmologist, where in the eye do you find the tapetum? Upper or lower? Upper. And why is that? The tapetum is an adaptation for dim light vision.
Light coming from below is hitting the tapetum, is going into the eye and hitting the top part of the fundus.
Okay, and what's special about the light coming from below as opposed to the light coming from above?
Darker?
Yeah, it's darker. It's just imagine an animal at dawn or dusk, and it's standing on a rock and looking out over the horizon. There's going to be a dark field, and there's going to be a bright field, and the bright field is going to be above, and the dark field is going to be below. So you want to be able to see that dark field. If you're a horse, you want to know where to put your feet. If you're a coyote, you're going to want to know where the mice are scampering around, but the bright light is going to be coming from above. So you want your tapetum to be in the upper portion of the eye. Now, what about if you're either a whale or a sea lion? The whale is an ungulate. Believe it or not, the whale is related to the even-toed ungulates. Cows is the most closely related land mammal. Is a hippopotamus. So actually the what's the clade of the even-toed ungulates? The parasidactyls is now called the sit parasidactyls to incorporate the whales as well, the cetaceans. So the whale is a ungulate, and the seals and sea lions are carnivores. So they're going to have two different kinds of tapetum, but where is the tapetum going to be located in the eye? It's a trick question.
Darker from.
I'm sorry?
It's still darker from below, though.
Yeah, it's the question. The answer was it's still darker, and that's pretty much the answer. They both hunt deep in the water, and so they want to be able to have that dim light vision, and there's no horizon deep in the water. So they both, each of them has a tapetum which covers the whole back of the fundus. One of the whales have a fibrous tapetum, and the sea lions have a cellular tapetum. So now you understand everything about the tapetum. You have to go?
Yes, I was the one here. Thank you.
So we talked about mammals, generally speaking, have eyes that are adapted for visual sensitivity, and reptiles, including birds, eyes that are adapted for visual acuity. Now I'm going to ask how did that come to be, evolutionarily speaking? We know that birds basically are reptiles. Whales are even-toed ungulates and birds are reptiles. There's no getting around that. So if you look into the paleontology of this, paleontologists believe, going back to the most primitive mammals, looking at the fossils, that they can identify features of the mammalian branch long before they became officially mammals, that already they have features that suggest that they're adapted for cool temperatures and nocturnal environment. They have hair, they have whiskers, they have, I can't remember what exactly the features are, but there's pretty large amount of paleontologic reasoning that suggests that even the most primitive mammals are adapted for cool environment. And in addition to that, they're already homothermic, they're already warm-blooded. So that warm-bloodedness helps them survive in the coolness of the night. And it gives them that niche, that nocturnal niche to survive. Reptiles, the group of vertebrates that became reptiles, are cold-blooded. And so because they're cold-blooded, they need to be in the bright sunlight in order for their metabolism to work. So if you need to be in the bright sunlight and you're going to develop, your vision is going to develop alongside that, you're probably going to develop vision that takes advantage of all that bright light and goes in the direction of visual acuity. So already, before dinosaurs, there's good reason to believe that mammals are warm-blooded and are adapted for the nocturnal lifestyle, and quite likely then have eyes that are adapted for visual sensitivity as opposed to visual acuity. Now, what's it like to be a mammal? One of those mammals that already is adapted for a nocturnal lifestyle and to try and stay alive during dinosaur era. What are some adaptations that such mammals would find beneficial? One would be to double down on the nocturnality. People use the term the nocturnal bottleneck to describe the situation of mammals that had to survive during the dinosaur era. I think that in paleontology, that term is not in as much favor as it used to be, but I still like it as one of the explanations of how mammals survived dinosaur predation. What are some other possibilities?
Living underground.
Living underground, living in burrows would be another good possibility. Living in trees would be another possibility, and that I like the idea that that's where our primates came from. Being teeny tiny in size is another possibility. You can scurry under rocks and find shelter, and probably not be as tempting to a big Tyrannosaurus Rex as a pig-sized mammal would be. So those are some features of mammals that lived alongside with dinosaurs. It used to be thought that there were not very many mammals, and when the dinosaurs disappeared, everything re-evolved from a small number of mammals. But that's no longer thought. If you look at the genetics of mammals, the common ancestor of various kinds of mammals goes way back into the Cretaceous or even the Jurassic period. So it's quite possible that many of the different kinds of mammals that we see alive today already were differentiated during the dinosaur era. It's just that they weren't differentiated very much in terms of what their fossils look like. They all look like mice or shrews. But when the dinosaurs disappeared, and these mammals that were already adapted to survive as being omnivores or opportunists or living underground, and being tiny, maybe eating seeds. Vegetative matter disappeared, but seeds were still available. Might perhaps have been selected to be the ones that survive, and then to go on with when there's no longer dinosaur predation to worry about, they go on to evolve to fill in various niches. Now, although they've done that, we have mammals like primates that have adapted to have impressive degree of visual acuity. The characteristic mammal is still a dim light specialist, a a crepuscular specialist, and the birds and reptiles are visual acuity specialists. If you're a naturalist and you're going to take a guided tour to find mammals, you're probably the best time to do it would be dawn or dusk. That's when probably the most mammals would be active. Birds, although if you're intending to record bird songs, then dawn might be a good time. But if you want to see bird activity, then during the daytime would be the best. Now, there's nocturnal birds for sure, and there's extremely diurnal mammals. So over time, things have spread apart. Now, birds are the same thing. Birds derived from theropod dinosaurs, but there are very few of them that survived through the Cretaceous extinction event. So they had to repopulate the world. But they're already exquisitely diurnal, exquisitely visual acuity. And so most of the birds adapted that in that way. So I'm going to put forward that mammals, as they exist today, have lost structural sophistication of their eye. Cartilage, we've talked about several of these already. Cartilage in the eye, shading in the photoreceptors, oil droplets, double cones, and there may be another one. Those are all things that are extremely common in reptiles, amphibians, fish, and largely absent in mammals. So I think they've been lost. I think they should, according to my way of dealing with these things by logic, they should have been present, and now they're not present. What kind of mammal would you look at in order to help to answer that question? Did they lose those functions, or did they just never have them? One thing would be to look at an amphibian, from which both reptiles and mammals derived, and we'll do that in a little bit. But first, we're going to choose a mammal.
Monotremes?
Monotremes, yeah. We're going to look at them. We're going to find the most primitive mammal that exists. So let's look at a duckbill platypus. Here's the duckbill platypus eye. Already you can see it's got a very robust cartilage in its sclera. It's got this phenomenon of shading of the photoreceptors. You're going to have to take my word for it. We use this very specimen using oil immersion to search carefully in the photoreceptors, and we found oil droplets and double cones. So they're both present in the monotreme. There's no annular pad in the lens. There's smooth muscle and not skeletal muscle. There's no conus papillaris or pecten. So I think one can guess that, yes, those functions have been lost. if you think about a duck-billed platypus, they're nocturnal. They come out and hunt in the daytime. They live in little rivers in Western Australia. Yeah, I'm sorry, Eastern Australia. And they during the daytime they go into a deep burrow and spend their daytime in a deep burrow. So if that's the way they were during the dinosaur era, I think a duck-billed platypus in the Cretaceous could have lived its whole life without ever having seen a dinosaur. So it makes sense that they were able to maintain their primitive form. Now the echidna, the fossils of echidna date from the Cenozoic, after the dinosaurs disappeared. And here's an echidna eye, also an egg-laying mammal. They also have cartilage in their eye. They have shading in the photoreceptors. And we have done plastic sections and electron microscopy on the echidna retina. And you can also see it, look in a book and find the same information. They do not have oil droplets and they do not have double cones. So they already have a retina that's a little bit more adapted like a typical mammal. And I'd love to know exactly what that means in terms of the evolution of mammals beyond the duck-billed platypus.
Can I ask a question before you move it? How
Yep.
can you tell that the RPE does or does not shade the photoreceptors when it looks like this? Like, what are you looking at that says like, oh yeah, that's RPE that shades the photoreceptors?
Well, look at the way it is. It's, oops. The pigment extends forward. Let's see if we can find some outer segments embedded in that. Let's go back to the eye. So we do have several echidna eyes. This one has no area where the retina is attached. I'm pretty sure we've addressed that.
Okay.
So cartilage, oil droplets. Oh no, the echidna doesn't have oil droplets, but the platypus has oil droplets, double cones. I can't remember what the other one is. So the next question is, let's look at an amphibian because the animals that eventually became mammals and the animals that eventually became reptiles and birds both split off in the Carboniferous from something that's like a lungfish that crawled onto the land and then eventually evolved into something like a amphibian. So I'm going to show you an amphibian eye. Thought I had something else.
The platypus is a bacromat.
Guess what?
So it's bacromat, like most mammals.
Oh,
Or all droplets, does that do anything?
that's a good question. That's a good question. I don't know the answer to that question. And it may have been studied. I know if you go, so birds and reptiles are, for the most part, tetrachromates, although that varies. It varies an awful lot as you go through the animal groups. But even the most, I'm going to show you a lamprey and a hagfish. Even the lamprey are tetrachromates. So tetrachromacy is not something that's evolved along with visual acuity. But so I don't know about the duck-billed platypus. I wouldn't think. I mean, they live either in the dark of a burrow, or when they're hunting, they shut their eyes.
All droplets, it's not.
Oh, that's a good question. Why'd you keep your oil droplets? That's a great question. I don't think we addressed that one. Carolyn Zeiss used this specimen to write a paper about the morphology of the platypus retina. So this is I'm going to show you the whole thing here. And I think you can easily tell what kind of an animal this is. Believe it or not, this is a frog. Frogs are like Volkswagens. They're very simple and reduced in there. So I'm not going to go into very much detail about frog eye anatomy, just to point out that they do have cartilage in the sclera. They do not have bone. They have shading of the photoreceptors, quite nicely just shown here. They have oil droplets. The best example that we have is actually in a tadpole eye. And they have double cones. They have smooth muscle, not skeletal muscle. They have no annular pad. So pretty much all the things that the duckbill platypus has, but many of which have been lost in extant either marsupials or placental mammals today. This is an axolotl, a salamander-like amphibian. Here's the better eye. So cartilage. It's not very clear that they have shading of the photoreceptors. This is an Alcyon blue PAS stain. They have nice, I believe that's an oil droplet there. And that's probably an oil droplet there. Notice how thin the outer nuclear layer is. And this is why I think that the schematic about identifying nocturnal versus diurnal differentiation by looking just at the retina doesn't hold so much for amphibians and reptiles. We'll come back to this when we talk about geckos, but a lot of nocturnal reptiles have derived their nocturnality not by having mammal-like rods, but by having repurposed cones that now can register in dim light. And we'll see how that plays out when we have I have a gecko. Most geckos are nocturnal. What time is it? Oh, we have about 15 more minutes. Thought I had pulled a lungfish, but I don't see it. Okay, well, that excellent. The eye of the axolotl morphologically is a lot like the eye of a lungfish, and we have the only lungfish we have that are still alive today are, I think, 3 different species. I think we have eyes from all 3 of them, and they look a lot like that axolotl eye. And I'm not totally sure where to go with that. But I'm going to jump now to, we're going to go through a variety of, we have 15 more minutes, so we'll get to some of them. We're going to jump through a variety of reptile eyes. And we're going to start with snakes. We mentioned snakes several times because they're paired with mammals. Snakes, unlike other reptiles, don't have cartilage. Unlike other reptiles, they don't have shading in the photoreceptors. They don't have an annular pad on their lens. They don't have a scleral ossicle, a bone in their eye. All of those features they share with mammals. They do have skeletal muscle, and embryologically or genetically, they pair pretty closely. Their last common ancestor is not that far away from the lizards, the iguanas and the chameleons and the geckos and the dragons, etc. So they're categorized as that sort of reptile. So what's going on here? Why do snakes and mammals? Why do snakes share those adaptations of the eye that are less associated with visual acuity alongside mammals?
Because they use other senses to hunt.
They do use their tongue. They stick their tongue out, and then it's got chemoreceptors. Some snakes have infrared light detectors, which gives them kind of a primitive view of heat. I don't know. I don't know about hearing in snakes.
Mammals.
They what?
They hunt mammals.
Some snakes hunt mammals. Yeah, many snakes hunt mammals. Yeah. So think about survival.
They hide in.
Yeah, so there's two theories about the evolution of snakes. One is, the one that I prefer is that they come from burrowing animals. They come from underground, and so they lived for a period of time, probably during the dinosaur era, not coming out, existing pretty much entirely underground, eating grubs and things like that. The other theory is that they have derived from aquatic animals, and that doesn't explain anything to me. I don't like that theory, and I think the common thought is shifting more towards burrowing animals as well. So let's preface that before we get into snakes. Let's preface that by looking at a burrowing. I have to go to the next. A burrowing mammal. This is a mole. In particular, it's an eastern mole. And the eastern mole has eyes, but they're covered over by skin. So it has anatomically cryptophthalmic eyes. In other words, the eyes have been pulled back into the skull. So there's no vision, of course. And this is the head of an eastern mole. This is the tongue of an eastern mole. And here's the nasal cavity of an eastern mole. And here's the eye of an eastern mole. And I think there's one on the other side. Here's the eye on the other side. Now, this is not a perfect section because here's the conjunctiva. The conjunctiva goes all the way around the eye. So obviously this is not sectioning every part of the eye, but let's look at what we do have. We have lens tissue. Look like a lens, look like a cataract, but there's no lens. It's not formed into a lens. It's just lens tissue. We have retinal tissue, but it's very primitive in the way it's formed. And so again, here is an animal living underground, and it's well on its way to losing ocular structures, as I propose that that mammals did during the dinosaur era, or snakes have done during the dinosaur era. If this animal continues to live underground or lives in a cave, it can lose its eyes altogether. And we have some examples of that. I don't know if we'll have time to show them. So let's look at a snake eye. How much time do we have? I think I'd like to have more time to look at snakes. So let's,
Sorry.
and before we go into a hodgepodge of reptiles, let's see what this is. That's a chameleon. Well, let's go back even further. Well, let's look at a shark. We haven't really, we looked at a zebrafish, and we're going to come back and look at more fish as time goes on. But now we're going to switch over in the evolution. We're going to switch over the fish and go to a more primitive era, and we're going to look at sharks. So this is most of the shark. We have a lot of shark slides. Most of them are pretty rotten. Some of them come from beached animals that are totally rotten. Some of them are a little bit less rotten, but this one's in good shape. The trouble with this one is it's a baby shark. So some of the features we're going to see here are developmental, and particularly look at the retina. So there's no shading of the photoreceptors, and I have to rely on memory rather than the actual experience. I don't think they have oil droplets. They have cartilage. They don't have bone. They don't have skeletal muscle in their eye. They have a lens that looks pretty much like a mammalian lens. There's no annular pad or anything like that. They have an exquisitely round lens, and I think I told you that exquisitely round lens, like a marble, is an indication of underwater vision. And when sharks were around, there was nothing but underwater vision. Sharks are able to accommodate to some extent because they can move their lens forward, and they have a muscle which is called a protractor lentis muscle, which I've never been able to find, so I'm not going to show it to you. Fish have a retractor lentis muscle, which I will show you, that pulls the lens backward in order to accommodate. So the reason why I'm fascinated by this shark specimen, my other interest as I became a specialized pathologist was in teeth. And so there's a question as to whether or not teeth, the first of the vertebrates that had teeth as we know them today were the sharks. And I'm going to show you the skin of a shark. And the skin of the shark is laden with little teeth, little dental-like structures called denticles. Have you ever gone to not the petting zoo, but if you go to the aquarium, there's a shallow, and you can touch either a ray or a shark as it swims by. It feels like sandpaper, and that's because they have these little denticles in their skin. So the question is, and I don't think it's been resolved, as to whether teeth differentiated from skin, or denticles in the skin differentiated from teeth, and that's an unresolved question. I like to think of teeth having come from skin.
Makes more sense, because I mean, the inside of the mouth is lined by
Skin.
skin.
Yeah, that's what I thought. Sharks are cartilaginous fish. They don't fossilize very well, but these little denticles fill up the strata where you find fossils by the millions. I find this hard to believe, but paleontologists feel that they can categorize different kinds of sharks by the appearance of these little denticles. And I think that pretty much takes us to where we want to end today. And we're going to pick up by going through a potpourri of reptiles. So if you can think of some reptiles you want to look at, go ahead and say it tomorrow, but we're gonna look at
Thursday.
what?
Thursday.
Thursday. Right, it's all tomorrow to me.
We have 2 meetings on Thursday, one, is there one in the evening and also, did I get some screwed up in my emails?